Calcein. Calcein, also known as fluorescent complex, is a fluorescent dye with excitation and emission wavelengths of 495/515 nm, respectively, and has the appearance of orange crystals. Calcein self-quenches at concentrations above 70 m and is commonly used as an indicator of lipid vesicle leakage. It is also used traditionally as a complexometric indicator for titration of calcium ions with EDTA and for fluorometric determination of calcium. Applications. The non-fluorescent acetone thuxidovate of calcein AM amplitude modulation acetoxymethyl is used in biology as it can be transported through the cellular membrane into live cells, which makes it useful for testing of cell viability and for short-term labeling of cells. Alternatively, furominous-2, Furaptor, Indominus-1 and ecorin may be used. An acetone thuxide group obscures the part of the molecule that chelates Co2, Mg2, Cn2 and other ions. After transport into the cells, intracellular esterases remove the acetone thuxide group. The molecule gets trapped inside and gives out strong green fluorescence. As dead cells lack active esterases, only live cells are labeled and counted by flow cytometry. Calcein is now rarely used as a Co2 or Mg2 indicator because its fluorescence is directly sensitive to these ions only at strongly alkaline pH, and thus it is not particularly useful for measuring Co2 or Mg2 in cells. Fluorescence of calcein is quenched strongly by Co2, Ni2 and Q2 and appreciably by Phe3 and Mn2 at physiological pH. This fluorescence quenching response can be exploited for detecting the opening of the mitochondrial permeability transition port MPTP and for measuring cell volume changes. Calcein is commonly used for cell tracing and in studies of endocytosis, cell migration, and gap junctions. The acetoxymethyl ester of calcein is also used to detect drug interactions with multidrug resistance proteins, ABC transporters, ATP binding, cassette transported genes in intact cells, as it is an excellent substrate of the multidrug resistance transporter 1 and D-RONP glycoprotein and a multidrug resistance associated protein mistopone. The calcein amplitude modulation assay can be used as a model for drug-drug interactions, for screening transporter substrates and or inhibitors, and also to determine in vitro drug resistance of cells, including samples from patients. Calcein is also used for marking freshly hatched fish and for labeling of burns in live animals.